Yeah, so for me, the clock now says that it's 5 past, so we waited a few minutes until everybody has joined us. Hello, everybody, and welcome to the second meetup of our series iGEM Phototrophs Overgrow the World. The topic for today is cloning methods and part standardization. And I'm Paul from the Bielefeld team, and together with Yazoo from the Marburg team, we will moderate this event. This series of events is a result of the partnership between our teams to create a community of teams working with phototrophic organisms in iGEM. We decided to dedicate this meetup to cloning methods and part standardization because we received feedback that you want to learn more about cloning methods. And for today, we invited several guests. At first, we have René Inckemann with a webinar about cloning methods. After this, we invited two people who developed recent cloning methods, Alejandra Schiavon Osorio, I hope I pronounce it correctly, for CyanoGate, and Andreas Andreou for the Mobius assembly. After these talks, we want to go over our plans for the iGEM Phototrophs handbook and how you can contribute to it. Then we take a short break and continue with breakout sessions. Like last times, in the breakout sessions, we will split up in 3 different sessions, one for cyanobacteria, one for higher plants, and one for algae. In these sessions, you will find experts working with these organisms on a daily basis, and they are eager to answer questions and help with issues on your project. After the breakout sessions, we planned a general troubleshooting session. And for those who are fit enough after all of this, we host a game night in the end. And now, without further delays, we start with a cloning webinar from René Inckemann, who is the PI of the Marburg team, member of the iGEM engineering committee, and who is currently working with Chlamydomonas in the Max Planck Institute for Terrestrial Microbiology.
Yeah, thank you for that kind introduction, and hello everyone from my side. I will try to share my screen. I hope you can see it. Does it work? Perfect. So yeah, again, hello everyone. So I want to give you some insights into like standardized cloning methods and especially modular cloning. And I've been using a similar presentation, or I gave this talk already in form of a webinar, and therefore we thought this might make sense to present to you guys. And if you have any questions, I'm happy to answer them in the end. So just a brief overview, what would we cover here in this talk? It's why using Golden Gate or modular cloning, and what the applications are, what has been done before, and what the basic principle of it is. Then like the basic biology behind it, the 101 of Golden Gate, and how this different level cloning works. And in the end, we come to really the practical part, how it would work in your lab or in your project. So first of all, I would like to start with like why this matters to you, or like why this might be relevant to you. And this kind of approach of this modular cloning and or this Golden Gate cloning makes especially sense if you want to assemble multi-gene constructs or genetic circuits or metabolic engineering, where you have to assemble several genes at the same time. Also, the advantage of that is that you can, for example, has been shown that you can assemble 24 parts in a one-pot reaction, which is very, very superior to other existing cloning methods. Also, if you really follow this kind of part-based cloning, sequencing is not necessarily required for like these higher assemblies, or if you want to do like huge libraries. And also you save a lot of time because you don't need to order primers for every of your designs. But if this doesn't make sense to you, you will understand in the end. And another big advantage is you can automate this method way easier than other cloning methods. So where did it start? So this modular cloning started in 2011. Back then, it was not highly adapted in the beginning because it was quite complicated to adapt this. But like then, like this system or this standard has been adapted to many organisms, not just Phototrophs. Just to use this also to advertise some work here. This is from a paper which resulted from our iGEM project in 2018, where we developed a modular cloning system based on this standard here for Vibrio nitrogens. But this kind of tradition was continued in Marburg since then, and we adapted this also for Cyanobacteria, or I'm also adapting this for the chloroplast of Chlamydomonas. So we really built up like a huge toolbox here in Marburg with that. But especially focus, or like especially attention, got this modular cloning in the phototroph field. So this has really been adapted for many chassis, and there's also like the Open Plant initiative, where all these toolkits are collected kind of. And just to name a few examples here, so there's, for example, the modular cloning toolkit for Chlamydomonas, for the nuclear engineering, and we will also hear later on about the CyanoGate toolbox, which is also following the general syntax of that standard. So why does it matter, or like why is it important to have like a common standard? So there was, for example, also like this kind of white paper, so to speak, where like many authors like wrote down this kind of standard for plant synthetic biology and agreed on that, and therefore it's quite easy to exchange parts between different labs. And also in the iGEM context, here on the right, this standard from that white paper has been kind of adapted. So you can submit your parts in that standard. This is not just something from the academic world and from the normal lab world, but this also has already been adapted in the iGEM context, not just by Marburg, but also many other teams. For example, the Kaiserslautern team in 2019 did it for Chlamydomonas. The Sorbonne team is using it since, I think, 2018. And Marburg, for example, also extended it for Cyanobacteria. Yeah, so just to mention the approach here, or like there's actually two approaches for the modular cloning, and I will rather focus on the left one, but I still want to mention the right one. So there's, on the left one, there's this hierarchical approach of like building up complexity, where every step involves like many part assemblies, and then you have this for example, examples like the loop assembly, where every time just combine two units kind of to assemble higher assemblies. So the right one has the advantage that it's way easier to adapt in the beginning, but it takes way more time for like establishing whatever, like a 5-gene, 10-gene construct. The hierarchical approach on the left is a little bit harder to establish in the beginning, because it's quite complex, but therefore you have the full flexibility when it comes to like multi-gene constructs. And also like it's quite fast to assemble construct like that. So what is the principle of all of that I talked before? So basically you like try to forget about this like classical vector insert thinking, that you have like a vector and you insert your gene of interest and then it's done, but rather like divide your whole plasmid or your whole construct you have in mind in like single parts, which are called level 0 parts. For example, promoters would be a level 0 part, RBS, your gene of interest, whatever, and they are stored in level 0 acceptor vectors. Then you can combine all of these in one pod and assemble them with one restriction enzyme to a single transcriptional unit. And then you can combine these level 1 transcriptional unit plasmids to combine them in a level 2 multi-gene construct. So you build up the complexity in each step from parts to one transcriptional unit to multi-gene plasmids. Like, how does it work exactly? So the classical restriction enzymes you might know from your lectures are these like type 2 enzymes. So they have a recognition site and they cut inside the recognition site. But in Golden Gate cloning, or in this modular cloning, this works a little bit different. There you are using the type 2S enzymes, which have a recognition site at the one hand, and on the other hand, they have a cutting site which is outside of the recognition sequence. So you can cut any sequence of interest. So these ends can be like any base pairs, and therefore you can choose them by design what these cutting sites would be. So you are not limited anymore to the recognition site of the restriction enzyme. And you have different enzymes. For example, BSA1 and BSMB1 are quite common enzymes in the Golden Gate or in the modular cloning systems. And this allows you a little bit of flexibility to build up these complex systems. So in general, when you are using these type 2S restriction enzymes, you have one special feature. So every time you cut, and if the recognition site is at the correct site, then you lose the recognition site after cutting. And if you imagine this like mirrored on the other side and ligated, there's no way that you can cut at that site again. So basically, after the assembly, there's no type 2S cutting site left over. So you cannot like cut it open again at that site. There is, of course, a special case if you would like switch around the recognition site and cut outside. You can still have this, and this also has some applications. But like the general case you have to remember is, for that assembly, you lose the recognition site, and therefore, yeah, this enzyme cutting site is not included anymore. And therefore, as I said, you can choose these N base pairs after your type 2S restrictions recognition site, and therefore people agreed on like a common standard which for base pairs you would have for different positions in a plasmid. So for example, a promoter would have GGAG on the left side of every part after it's cut by the type 2S restriction enzyme, and the same is on the other border. And therefore you can use one restriction enzyme for like many plasmids you throw in on many sticky ends you want to create, because you always have different 4 base pair overhangs which are created by this type 2S enzyme. And this looks a little bit overwhelming in the beginning, but in the end, you can just like build up your own kind of like toolkit where you use these different positions which are standardized, and then you just have to connect from left to right to assemble a full plasmid. So for example, promoter, RBS, coding sequence terminator, and so on. And then in the end, it ends up as a full plasmid for like one transcription unit. So let's go a little bit deeper into like how this usually works. And this is just one example. Of course, like the different toolkits differ a little bit in the different assembly steps, but like in general, you can still learn something if I explain you this example here. And we have in the phototrophic field, we have this universal acceptor vector, which is also standardized, which is also a derivative of the iGEM standard vector for biobricks. And This vector contains a chloromphenical resistance and a high copy origin. And in case of our Marburg collection here, we have like a full expressing GFP in there. And this has the function that if you replace this one with your part of interest for your basic part, let's say you would like to introduce a new promoter to your system, then you would cut out the GFP and put your promoter in there, and you can select for that in the end by having green and white colonies in case of your E. coli transformation. And then you store your basic parts, your promoters, RBS, your gene of interest in this level 0 acceptor vector. Then you take like many different level 0 acceptor or level 0 parts, and then you can assemble a full transcription unit in level 1, as I mentioned. So, for example, you would pick a promoter, you would pick a 5' UTR, you would pick your coding sequence and assemble it into a level 1 plasmid, or like assemble it with resistance and ORI. And in this case, we are switching the antibiotic resistance in order to get rid of all the remaining not assembled level 0 parts. And we also have like a visual marker in this case, which is a red expressing cassette. So basically you can then select on red and white colonies. And therefore you just like put all your parts together, and in the end will be assembled to one transcription unit. Before I come to level 2, I want to make like a small side note here, which is library cloning. So let's say you want to test a certain promoter or a certain like enzyme where you have like many options to choose. Yeah, you don't want to clone them like separately completely. So there you have the possibility in some systems of putting a placeholder in first place in like these higher assemblies. And this placeholder can be cut out again, and you can put like your whole library of promoters, of coding sequences or whatever in there, and therefore test them all with a really, really easy cloning step, which is just 2 parts. But this is something, if you have questions about it, you can ask me later, but I don't want to go in detail here how this works in detail on a molecular level. But let's say you want to express not just one enzyme or like one gene, but you want to do many gene constructs, and therefore you can do the level 2 assembly. In some systems, it's also called level M. And then you have different systems, how they connect, either with backbones or different backbones or different connectors. Then can combine your different level 1 plasmids or transcription units to higher assemblies, and you predefine in the level 1 how they are assembled in the level 2. And in some systems, for example, in the system we have here in Marburg, we can even switch the direction of your transcription unit, which has some advantages in bacterial systems. Yeah. So coming back to the overview, so basically, if you would like to use that, you would kind of get maybe an already existing toolkit with level 0 parts, which already contains some promoters, RBS, and so on. And you would implement your genes of interest or your parts of interest into also level 0 ones. It's also called domestication sometimes. And then you would use these level 0s, put them in level 1, and can use that for level 2. That's kind of the cloning workflow you will always follow for using this modular cloning system. So to make this a little bit more practical, or like how this would work in the lab and how it differs from the classical restriction ligation methods you might know from some practical courses or whatever. So in fact, you're really pipetting all the plasmids in one tube, and you also like pipette the enzyme and the ligase in the same tube. So you don't do different steps for like restriction and that ligation, but rather put it in a thermocycler And there we have this protocol, or like also different protocols for different complexity assemblies, where you switch between an optimal temperature for restriction and ligation and cycle this every time. And due to the fact that you lose the recognition site for the cutting site every time, every cycle you increase the chance that you have full assemblies and correct assemblies in your tube. In the end, we just like do a final digestion, we call it, and this is to like cut open all the remaining parts which are not circularized to avoid to have too much background on your coli plate, and in the end we just like inactivate our enzymes. So basically it's really like pipetting everything together, putting it into the thermocycler and running this reaction. And you don't have like a shitty gel extraction in between. You don't have like dephosphorylation or so. So this is really an easy method to implement in any lab. And the transformation is basically exactly the same like any other cloning method. I just wanted to highlight the possibility of like color selection with, for example, the GFP and the RP cassette we are using in some systems. There's also LUX-set, of course, which can be used for like differentiating between successful assemblies. Just a few words, but not too much detail about like how you can use automation for this. So for, there's like many different robots, of course, and some iGEM teams might have the chance to have some access. And there's, for example, the OpenTron, which was sponsored to some teams in 2018 and 2019. And there you can also like use that, for example, doing all the combinations of level 0 combining and like doing the assembly in the OpenTron. There's another machine which is a little bit more fancy and a little bit more expensive, but super cool for that Golden Gate method, and that's the Echo. And it's an acoustic liquid handler, so you don't need pipette tips anymore. I don't want to explain how this works. It's super cool. I can highly recommend to Google this and like see how this works. But there you can even scale down your Golden Gate reaction to a total volume of 1 microliter or so, and therefore save a lot of reaction costs. So, and do like whatever, like 96 different constructs in like a few minutes. So yeah, automation is really easy with Golden Gate assembly. And with that, I'm happy to take any questions.
Thank you very much, René, for your presentation.
You're welcome.
Questions can also be written in the chat, and then I will read them to René.
Yep.
Okay, at least there's from Mathilde Bier, I think. It was super clear, thank you. So maybe the advice, it was so good, there is no need for questions.
I had a...
Yeah.
Can I ask a question about the...
Of course.
Well, just so thank you for your presentation. It was very clear, but I had a question about the library cloning.
Mm-hmm.
Could you explain with a little bit more details about the this technique for choosing like a promoter or something?
Yeah. So so I explained a little bit before here that you can put the recognition site for a part in the other way around, that you still have the recognition site. So what we did here is putting a GFP expression cassette for like a promoter position, for example, or so, or like any enzyme position, and putting, instead of like the final promoter, putting this GFP cassette in there with the inverted recognition site for the assembly. Therefore, we can like cut out this GFP cassette again in the next cloning step, and then put our whole library. So what we would do is first would assemble like the full construct we want to build, and just insert the placeholder before, and then it's just basically one big construct you build, and afterwards exchange that and put all of your library in there. You can even, like in some cases, people even combine all of the library in one tube and just do one assembly afterwards. But you can also do it separately. But like this is really efficient because you can also put them just in one tube, and in the end just pick many colonies. That's how we use it here. I know it's quite complicated if you don't have the exact sequences, but like if you have further questions, I can still also help you with that.
So if I understand well, the promoter, if you construct this construction level 1 by an enzyme like BSA1, to remove the GFP in position 1,4, you have to use another restriction enzyme, right?
You could do that, but in our case, we actually use the BSA1 again, although it is not really following the Golden Gate kind of rules, because then we still have the recognition site in there. That's true. And this also causes a little bit of problems. But if you have as a final step a ligation, you can still have this recognition site in the end and keep it in there in the final construct. And you can also like screen in the beginning then for our GFP. And then use BSA again. BSA again to cut this out.
Okay,
BSA once. Yeah.
Okay, that's interesting. Thank you.
No problem.
I have a question. So is there a size limit for the vectors you create?
Actually, no. So the size limit is rather given by E. coli. So there has been a paper this year where NEB, for example, assembled a 60,000 base pair construct with that method in one pot, like 50 parts together, and assembled a whole phage genome in one reaction. And I would say, if you want to exceed that, have fun.
Yeah, but I mean, 60 kb is already bigger than anything I imagined before. So
yeah, so, and it was still not at the full limit. They still found a lot of colonies having the full assembly in there.
And the construct was fully functional?
Yes, it created the fully functional phage.
Insane. Okay, yeah.
Any other question? Also, like if you have any other question which comes up in the next days, you can always write me on Slack or like join later on in the troubleshoot sessions also, and still can ask questions. So it's fine. Or if you encounter any problems within your project in the coming weeks, just yeah, let me know.
Yeah, so now we have a bit surprising experience that we are nearly 20 minutes ahead of our time schedule.
We could start with the next presentation. Do you think that's a good idea?
Okay. Yeah.
Yeah. Okay. So for our second presentation, we have as guest Alejandra Schiavon Osorio. She has done her undergraduate degree in biotechnology in the ITESM in Mexico, a master's in biotechnology from the University of Edinburgh, where she participated in the 2016 iGEM competition. There, the team worked in standardizing parts for non-modular organisms. Currently, she's finishing her PhD in Alistair McCormick's lab at Edinburgh University, and her main research interests are the expansion of available tools for cyanobacteria research and the implementation of such tools in non-modular cyanobacteria for the production of high-value compounds. Thank you very much for coming, Alejandra.
No, thank you very much for the invitation. Let me start sharing my screen. Cool. Can everybody see my screen? Yeah. Cool. Yeah, thank you very much for the invitation. I really appreciate it. As you mentioned, I did iGEM some years ago, so I always have this passion for sharing research in a non-traditional ways. So I found that these meetups are quite interesting and quite helpful for all the communities, and it is really catching up for this interest in the Phototrophs in the last 5-10 years in iGEM. So it's really good that people are getting interested in their capabilities, and my main interest is how we can push them forward so we can share parts between labs and everybody can start working with them and be more amenable for working. So as you know, cyanobacteria, they have this huge potential because they can transform CO2 with light using Rubisco and several metabolic pathways into very useful second high-value metabolites like chlorophyll, carotenoids, terpenoids, flavonoids, lignin. I work also with the production of phenolic compounds. There's a lot of interest in the pharmaceutical and cosmetics industry for these phenolic compounds. And compared with model plants, they are relatively faster growing. They have a bit more genetic trackability. I see them like this in-between point between bacteria and plants. They can do photosynthesis, but they are not as complex as algae, for example, or plants, but they are not as simple as an E. coli, and they have a lot of things to offer. Sadly, when we talk about synthetic biology in cyanobacteria, they do lag behind. In the last years, there has been a clear expansion of the parts that have been characterized. Now we have CRISPRi tools, we have more model parts being translated from E. coli to cyanobacteria. We have been working with non-model organisms in cyanobacteria that have fast growing, they're close to yeast. So there is room for improvement. And one of the things that we have found is that the parts from E. coli that we have translated to cyanobacterial research, they do behave different. So there's this need that we can have high throughput technologies for cloning, for example, so we can test more E. coli parts in cyanobacteria so we can see how they behave. So jumping into the cloning, so all of our Golden Gate system is based in the modular cloning toolbox for plants. So in 2015, the Nicola Patron lab developed a whole suite of backbones. So they have level 0 backbones for promoter, coding sequences, terminators, nuclearizing tags. A lot of things that are very specialized for plants, but we took the best of their toolkit and we adapted it. So they, as it was mentioned previously, it uses these hierarchical levels. So we're going from level 0 to level 2. And I never liked these diagrams. They tend to see very complex when we talk about cloning. And I think a similar thing was done before. Like, let's think as Golden Gate or genetics as if they were Legos. So we have level 0 parts, let's say a promoter. These are with the overhangs standardized with type 2S restriction enzymes that we have this selected from OpenPlan or iGEM, that are the phytobricks in this case. So you can have your library of level 0 parts, you can combine them together and assemble a level 1 expression cassette. So for level 1 in the MoClo toolkit, you have 7 different backbones because you can have 7 different positions later on. And very ad hoc with the Olympic Games, I decided to go with a cherry blossom. And then, so the jump from level 0 to level 1 is using BSA, and then your your jump from level 1 to, we introduce this new level in cyanobacteria called level T. So we call it level T for transformation. It is equivalent to a level 2, so it can accept multi-gene constructs. So you jump from level 1 to level T and BPI. And different to what you would normally do in E. coli, in cyanobacteria you can have two options for this part. You can go with chromosomal integration, so we have backbones for doing genetic modifications directly into the chromosome, or we have self-replicating plasmids that you can just express as you would normally do in E. coli. So we have adapted the Plant MoClo as a CyanoGate to do very different things. So you can assemble expression cassettes. I will talk much more in detail for people that are not specialized in cyanobacterial research. You can generate mark and unmarked knockout mutants. You can generate knock-in mutants as well. And we also have tools for gene silencing with CRISPRi. So for the characterization of synthetic promoters and native promoters, what we wanted to give the user is a wide range of promoter to choose. If they wanted to express something with a very strong promoter, then they would have an option. If they wanted something that is not very, that you would need to produce in specific amounts, low titers, that's what we wanted to give the user, a high dynamic range for their expression levels. So we took 30 heterologous and synthetic promoters, mostly from E. coli, and 13 native promoters from cyanobacteria. And we characterized them in 2 different organisms. So we decided to go for the model organism because most of the cyanobacterial research is still being done in Synecocystis 603. And at that moment, there was an excitement for this new strain called Synechococcus elongatus Utex 2973. It is a fast-growing species. Under certain conditions, it can grow as fast as yeast, so a doubling time of 2.5 hours, which is quite impressive for a cyanobacteria compared with 603 that is around 6-7 hours. So that it is a huge improvement, bringing closer cyanobacteria to like industrial purposes. So to characterize our promoters, we have our promoter, and we selected EYFP as our selected fluorophore and a terminator. We assemble everything in our PPMQ AK1 level T plasmid for conjugation into cyanobacteria. And this is what we got. I'm not going to go into details into which type of promoters were located where. So if you are familiar with a bit of iGEM, so we have the very strong J23119 promoter. So this is the strongest promoter that we found for cyanobacteria. And in relative terms, we have 120 dynamic fold of promoters that you could use for your cyanobacterial research. So this is quite cool, but I think our main development, so one thing that, as I mentioned before, that the parts behave different between cyanobacteria and E. coli, we found that they also behave slightly different between cyanobacterial species. So we have an R-squared correlation between 2 species of cyanobacteria of 0.84, which is good. Like if you have a part that has been characterized maybe in a model cyanobacterial species and you want to use it in a non-model species, maybe you would have an idea how the part will behave once you test it. And we also took native promoters from cyanobacteria. So the CPC560 is one of the strongest promoter in cyanobacteria. It codes for the phycocyanin operon, which is what gives cyanobacteria their bluish color. So we created different variants with the same purpose of creating a dynamic range of promoter options for the user. As a general structure of the promoter, it is divided It has transcription factors upstream, it has a middle region, and a downstream region. So we decided to make different variations, duplicating the downstream region, duplicating the upstream region, to see if the binding of more transcription factors to the promoter will increase its activity. But at the end we found that the native version was already very, very good one. So all the other versions, they just give a good dynamic range to the user. So the greatest achievement in our research was to adapt the CyanoGate MoClo toolkit from plants to make it amenable for cyanobacterial research, because in cyanobacteria you tend to do a lot of insertions or deletions in the genome, and having a standardized way to share parts or having easy to make constructs was a huge improvement. So cyanobacteria is very good at doing homologous recombination. A species like Cyanocystis 603 are naturally transformable, so they can take up DNA really easily. So you can just mix your DNA with your cyanobacteria, and the cyanobacteria will take it up. And for this, we develop a two-selection method, or a two-step selection method. So in a first step, we'll have a positive selection method with an antibiotic resistance cassette, and then later on a negative selection with the SACB gene. One thing is that cyanobacteria tends to have multiple copies of the genome depending on the growth state or conditions in the media. So it can go from like 10 copies to 50 or 60 copies of the genome. So for this purpose, you do need to ensure that your copy of your insert, it is present in all the copies of the genome, so you can progress to your research. So I will do a detailed explanation of how you do an integration via homologous recombination to generate an unmarked mutant in cyanobacteria. So first you will have to construct your plasmid. So this would be a level 1 plasmid. So in your promoter acceptor vector, you will clone your up flank region. So this is the homologous recombination arm. Then we already will provide you with these parts for the SACB, and the antibiotic resistance cassette. You can choose from several ones depending what you want to do. And your down flank will be cloned into the terminator acceptor, and then all of these come together in a level 1 plasmid. So then in your genome, you will have pretty much something similar. So you will have your up flank, that it's the same as this one, because this is where you want to delete your gene, and your down flank. And this is the gene that you would like to generate your knockout. So after you added your DNA to your cyanobacteria very nicely in the media, you just add your DNA. These two will recombine. And then you will select for your antibiotic resistance cassette to push that this homologous recombination happens. And you will end. So you will have deleted your gene, and you would end with copies in your genome of the SACB and the antibiotic resistance cassette. So this is very useful. You have deleted the gene of interest that you wanted, but let's say that you want to do several knockouts in different places of the genome. There are only certain amount of antibiotics that you could use. So if you wanted to do, let's say, 5 different deletions and you only have 4 antibiotic cassettes, you will have a problem. So what we want to do is like to get rid of this part in the middle, so you can either recycle this antibiotic resistance cassette for another deletion in your genome. So the way that you do it is quite similar. You will recycle your up flank from your level 0 part and your down flank. And now you will have an unmarked linker in between of them. So this is just a small piece of DNA. I think it's 27 or 28 base pairs, that it will just be like a placeholder just to close your plasmid. You will again combine it with your cyanobacteria in the liquid. homologous recombination will happen. But then the SACB gene is a negative selection mark, will work as a counter selection marker. So the SACB gene produces an enzyme, that in the presence of sucrose creates a toxic compound. So the cyanobacteria that doesn't undergo the homologous recombination and keep the copies of the SACB will die. And the nice ways to test it is that if you have this construct after your homologous recombination, your cyanobacteria will grow nicely on sucrose, but it will die on kanamycin. So that's the best way to check. And this is why the segregation is very important, because if you have one single copy left of your kanamycin resistance cassette, the colonies can still grow on the kanamycin and still survive on the sucrose if they have like a mixture of genome copies going on. And so this sounds very nice and in the theory. So in our toolkit, we provide the already pairs of flanks to do homologous recombination in neutral sites if you want to integrate anything. So in cyanobacteria, we call a neutral site a piece of DNA that has been already proved that if you delete that piece of DNA, the growth and the phenotype of the cyanobacteria appears to be unchanged. So that's what we call them, neutral sites. So far, there have been around 25 identified in 603. So we included pairs of flank for 4 of them. And we also included for other model species. So we included them for C7002, that is a marine species, and Utex 2973, and Synechococcus elongatus 7942. So these ones are very similar strains. We also provide 4 antibiotic resistance cassettes so you can choose from, and also we provide the SACB so you could do the counter selection. So if we try as a proof of concept of how the system works, we amplify the homologous recombination amp, so the up flank and the down flank for the CPCBA operon, as I mentioned before, is what gives cyanobacteria. So it produces the phycocyanin operon, so it's what gives the cyanobacteria their blue color. And we inserted the SACB antibiotic resistance gene, and so this process, it is quite demanding because you have to be sure that your genome is completely segregated, and also the CPC operon, it is part of the photosynthesis machinery in cyanobacteria. So it is something that is very slightly difficult to delete because the cyanobacteria will be pushed and have a limited capability to do photosynthesis. So you can spend between 3 and 6 months sometimes trying to delete genes in cyanobacteria in this part. And we decided to go for that gene because it was giving an obvious change in the phenotype after we have deleted our gene. But the good thing with the Unmarked system that we have is that the previous process took about 3 to 6 months, but then the counter selection to delete the SACB and the antibiotic can be as fast as 2 weeks. So here we can see that after the double homologous recombination event and selecting on sucrose, we have completely deleted the SACB and the antibiotic resistance cassette. So this is one option once you have a marked mutant. So you can generate an unmarked mutant of whatever you want to have. But the second option is that you could replace the antibiotic resistance cassette and the SACB with an expression cassette. Let's say in this case, we just wanted to insert an EYFP in our, what we call an olive mutant, so the deleted CPCBA operon. So this is a little bit more complex assembly. So you will have 3 separate level 1s. So you will assemble your cassette, your promoter, EYFP, terminator in the L1P2. And you will still use your same homologous recombination flanks that you have used before, but we have adapted these tiny pieces here, so you can use so you can assemble them in level 1 without having to generate a new level 1 part. And then all of these you will clone it into your level T for homologous recombination. And then instead of deleting your gene, you would have inserted in its position an EYFP cassette. So we have the marked mutant and then an unmarked EYFP cassette in the place of the CPCBA mutant operand. And we prove this after doing flow cytometry. So we have the wild type, it doesn't express any fluorescence. The olive, it doesn't express any fluorescence. And then now we have proof that we can do insertions with our system into the genome and replace the antibiotic and the SACP cassettes with, in this case, EYFP mutants very easily in less than 2 weeks. And another thing that you could do with the CyanoGate toolkit is using it for CRISPRi gene silencing. So as you know, in the CRISPRi system, so the dCas9 has been modified, so it doesn't have a catalytic activity. It will just bind to where it will recognize the sequence, and it will block the transcription to go forward. So the RNA polymerase will be stopped because it will found itself with the dCas9 in front of it. So in this case, to prove that we could assemble constructs that were able to do gene silencing, we took the previous CPCBA deleted mutant that has been replaced with the EYFP. So this is in the genome. So the EYFP has been expressed in the genome right now. And all the dCas9 and the guide RNAs are expressed in a self-replicating plasmid. Yeah. So here we have, so in one of the constructs, we have a promoter. So this promoter doesn't have a ribosome binding site, so it's just a transcriptional star promoter. You would be provided with in our toolkit, we provide the sgRNA scaffold. So here you would generate or design your guide RNA depending to what you want to silence. And then, so you will have, in this case, we did a strong promoter to express our guide RNA and a strong promoter to express our Cas9. So all of this is cloned in the same plasmid. And we decided to target the EYFP, the guide RNAs in different positions to see if there was an effect, or does it influences, like in the literature, it says that the efficacy of the silencing is more about where do you position your guide RNA in the gene, rather than the RNA being able to be stopped by the Cas9. And yeah, we did find that depending on the position where you put the dCas9, the expression of the fluorescent decreases up to 90. So we were able to generate a 90% silencing of the EYFP with our system. And you can see that when you have the guide RNAs by themselves without the Cas9, fluorescence is not affected. The olive mutant doesn't express any fluorescence. And this is the control of just nothing is being expressed in the olive background. So one other thing in cyanobacterial research, including the lack of tools, is that we do have a limited number of self-replicating backbones that we can use. So for example, in this previous example, we had to use the olive mutant with the CPC 560 expressing the EYFP in the genome, because at that point when we were doing the research, we only had one backbone to do the self-replicating. So it was being problematic to express the GFP in the self-replicating backbone as well with the dCas9 and the guide RNAs. It was like such a big plasmid, so complicated to be everything in the same origin of replication. So later on in the paper, what we did is looking for other origin of replications that could be useful, so for cyanobacteria. So we domesticated a PCVA 421 backbone that has an RK10 origin of replication instead of so the PPMQ has an RSF 1010 origin of replication. And we characterized this one, another nice thing, the PPMQ is a kanamycin backbone, and the PCVA is a spec backbone. So now we have two backbones with two different antibiotics, so you can use them in conjunction together. The RSF1010, sorry, the RK2 gave much lower fluorescence than the RSF1010 when they're expressing the same construct. So we decided to characterize the copy number of the plasmids, so we can explain why it was being much lower. So we found that the RK1010 is a very low copy number plasmid in cyanobacteria. So you only have between 7 to 10 copies of the plasmid in the backbone. So that is particularly useful, for example, when you're expressing toxic proteins like DCAS9. That has been shown that bacteria doesn't tend to like it very much when it has been expressed. So if you have it under a under a low expressing promoter and in a low copy number backbone, you might have better results in your research. So as a summary of like what's included in the CyanoGate toolkit, so we have it includes more than 50 promoters. It has, some of them are, we also included, we didn't do the characterization, but there are also native cyanobacterial inducible promoters in the toolkit. It has flanking regions for homologous recombination. So the linkers are in order to aid all the generation of the marked and non-marked mutant. We included 13 CDS1s with different purposes. So we included EYFP, we have the different antibiotic resistance cassette, the SACB. We included also 21 terminators that later on these terminators were characterized in a different publication. And we included 6 new acceptor vectors for different purposes in the toolkit. So in total, the toolkit is a quite wide range of tools that it includes. So in total, it's 116 parts. It took us more than 2 years to develop all the research and characterize everything and do the deposit in iGEM. So yeah, we are happy. If anybody is interested, please, we are very happy to share parts and backbones, and or if you want, you can get it all together in iGEM. But we understand that sometimes the iGEM teams are under tight budget, so we are happy to share if you're interested in any parts. Just you can drop us an email for that purpose. And so I just want to acknowledge all the people that were involved in these years in the project. So Alistair McCormick, my supervisor, Grant and Ravi. Ravi now is in Cambridge University. So the three of us, we like mostly developed the CyanoGate toolkit, and we have a lot of collaborators and a lot of sponsors that made the research possible. And I think that's pretty much it. So I'm happy to take any questions.
Thank you very much, Alejandra, for your presentation. Okay, Paul, do you have a question?
Yeah, I have a question. So first, thanks for the great presentation. And so I'm a bit wondering, or I didn't understand completely why it is necessary to exchange these marked things by these unmarked constructs.
It is not completely necessary. So once you have deleted your gene and you have inserted your SACB and your antibiotic resistance cassette, you can stay there. So you don't have to remove it. You don't have to make it unmarked. It's just, so we had conversations a bit with industry, and what they told us is like in the big scale, if they at some point deleted the gene or did insertions into the genome, it is not feasible in an industrial scale to keep running your mutant with antibiotics.
Ah, okay.
So that is one of the reasons, and also is there's a limited amount of antibiotics that work in cyanobacteria. So let's say that you wanted to do multiple deletions in the genome. Every time that you would do a deletion, you will have to replace the deletion with an antibiotic. So let's say if you wanted to do 5 or 6 simultaneous deletions in the genome, you would encounter a problem because you don't have that many available antibiotics to be selecting on.
Okay, yeah, no, I understand it. Thanks.
Yep.
So there's also a question from the chat from Likith Chandragiri. I hope I spelled your name correctly. The CyanoGate protocol for PPMQA-K1 mentions that the helper strain to be used for triparental mating is MC1061 E. coli strain containing vectors PRK24 and PRL528. Would it be possible to use E. coli HB101 with vectors PRL443 and PRL623 instead?
Yeah, they are like interchangeable. At the end, the function of that HB101 with the PRL443 and 623 is exactly the same as the one we use in the publication. So they can be interchangeable if you already have the other strain for your conjugation.
So thank you. René has a question.
Yeah, so thank you for your talk. I was wondering about your second last slide where you showed the RK2 ORI. Was this just tested in 6803 or also in other strains? Because as far as I know, like in other strains, it's even more issue with the replicating backbones, isn't it?
So at the time of the publication, we have only tested it in the RK2. But we have tested the RK2 in UTEX 2973, and it does replicate and gives like we just use the same one like just to express some fluorescence, and we did get fluorescence values. So the backbone does replicate in other cyanobacterial species, at least that we. So it would be in UTEX 2973 and UTEX. Sorry, in UTEX 2973 and Elongatus 7942. It would replicate in those two.
Nice. Okay, thank you.
So do we have any further questions? Okay, so I don't think we have. We are again a bit early for our next presentation.
Yeah, first the question to René, whether that hand is again up or still up.
Oh.
No, sorry, I didn't put it down, sorry.
Okay, so Andreas, we are way ahead our time schedule, but I see that you are already here. So would you like to start your presentation?
Yeah, sure.
Okay, it's my great pleasure to introduce Andreou to you. He got his bachelor in Athens in biology, and after that he moved to Edinburgh for his master in system and synthetic biology. And then he stayed in Edinburgh for his PhD in plant synthetic biology. And currently he's a postdoc in Edinburgh working on a novel protein purification system, and also on the enamel matrix proteins for the treatment of tooth decay. So in my very basic view, sounds not really plant related, but also very interesting. And his research interests fit perfectly in today's Meetup topic. And they focus on developing synthetic biology tools to confer complex but predictive genetic engineering in biological systems, and also to promote standardization in plant synthetic biology. And with this introduction, I hand over to Andreas for his talk about the Mobius assembly.
Can you see my slides? Everything is okay?
Yes.
Yeah. Yeah, thank you very much for the invitation. It's really nice to organize these meetups and exchange knowledge. Today I will talk you about Mobius assembly, which is a DNA assembly system I developed during my PhD, which I adopted for plant systems. And I will talk a bit for some like protocols and some parts I also developed for plants during my PhD. Even though now I'm not working with plants, I moved to like yeast and bacteria. I hope sometime in the future I will return back to plants because they are really cool systems. I will start with a small history. And the first attempt for standardization. This one is not like widely known. The first attempt to introduce standards in plasmid assembly was done like 25 years ago by Rembachuk with the system Nomad, the cloning system Nomad, which it was very similar to Golden Gate assembly. It was using type 2S restriction enzymes to assemble standardized built-in blocks and make reusable modules. And also it was allowing for sequential and directional insertion of any number of modules. This system, even though it was like really cool and cleverly in design, it wasn't adopted by the scientific community, probably because it was too early back then. But yeah, if like the scientific community was adopting this method, everything could have like become or develop much faster. And then a few years later, we have the introduction of the development of Golden Gate assembly, which everybody of you know, which again uses type 2 restriction enzymes. We have at the same time digestion and ligation of the products with the action of a restriction enzyme and the T4 ligase, and the first generation of DNA constructs. Even though Golden Gate assembly was a powerful method, it had a limitation. The final products were not reusable. This was changed in 2011 with the development of MoClo and Golden Bread, two methods that they introduce a second, and in the case of MoClo, a third restriction enzyme, and also an extra or two extra cloning levels. So the composite parts were like reusable. MoClo became like really popular, and most of the new developed assembly methods for different organisms, they are based on MoClo. I have a list here, but probably there are more. The characteristic of the strong characteristic of MoClo is the high capacity. So you can make large constructs really fast, but it has like complex toolkit. On the other hand, Golden Bread, it has low capacity, which is a limitation, but it has a simple toolkit. This was changed with the introduction of Mobius assembly and loop assembly, which they combine the benefits of both methods, high capacity and simplicity. Yeah, Mobius assembly It works in 2 levels for cloning, level 1 and level 2. Level 1 is the generation of transcriptional units, and level 2 of multi-transcriptional units. As I said before, it combines both cloning capacity with a simple vector toolkit. It has lower domestication requirements. It uses, along with BSA1, the rare cut AR1, and it uses visual cloning screening, and there is no need to use IPTNG or XCal to visualize the colonies, and also is compatible with existing libraries. So we have level 0 and 1 acceptor vector, as it happens most of the CyanoGate-based assembly methods nowadays. And then we have level 1 with 4 vectors, level 2 again with 4 vectors, and 7 auxiliary plasmids, which they help the generation, they help in the cloning of level 2. And from level 1 to level 2, it goes with the use of BSA1, and from level 2 to level 1 with AR1. As I said before, I replace the blue-white screening for colorful screening, and I use constitutive expression of chromoproteins and superfolded GFP, and the use of the avoidance of IPTNG and XCals, it makes the cloning less costly. And also it's really easy to distinguish between the different cloning levels. So if you see, for example, pink colonies, you know that you are in level 1. If you see yellow colonies, you know that it's level 2. Level 2. So in the Mobius acceptor vector, I'm using AmylCP, SPIS-ping in level 1, and superfolded GFP in level 2. However, now we're thinking to replace SPIS-ping with mRuby because SPIS-ping it has a bit of toxicity in the cells, and sometimes there is like difficulty reviving the cells from the glycerol stocks. So in level 0, we have the generation of the standard parts. The Mobius universal acceptor vector converts the DNA fragments into standard parts. As I said before, I use AR1 restriction enzyme. It recognizes 7 base pairs to cut. So there is a reduction of domestication requirements. And I follow the Phyton-Brick standard. As you know, the Phyton-Brick standard, it's the iGEM standard for type 2S restriction assembly. And the purpose of using this standard is to facilitate parts sharing. And what happened in level 1 assembly, we have the generation, the assembly of standard parts and the generation of transcriptional units. For example, here we have 3 Phyton bricks. That's for plant parts. We have a promoter, and gene, and a terminator. So we combine those 3 parts in a level 1 acceptor vector with the action of BSA1, and we get the transcriptional unit, promoter, gene, and terminator. And then we go to level 2 assembly, where we combine single transcriptional units into multiple transcriptional units to make a higher hierarchy constructs. And I have an example here for single transcriptional units, transcriptional unit 1, 2, 3, and 4. along with the auxiliary plasmid 4A. They combine in a level 2 acceptor vector, and they make a 4th transcriptional unit construct. And then if we go back again to level 1, actually here I explain how we use the auxiliary plasmids. There are 2 types of auxiliary plasmids, the 1, 2, 3, and then 4A, 4B, 4 gamma, and 4 delta. 1, 2, 3, we use them when we assemble less than 4 transcriptional units in level 2. For example, here, if we assemble 3 transcriptional units, we'll use the auxiliary plasmid number 3, which provides the overhangs necessary for the assembly. And then the other 4 auxiliary plasmids, we use them when we have the assembly of 4 transcriptional units, and the 4 letters correspond to the 4 vectors, alpha, beta, gamma, and delta. So in this case, because we are assembling 4 transcriptional units in vector A, we are using the auxiliary plasmid 4A. And then, as I was saying before, if we want to make a higher, larger constructs, we go back from level 2 to level 1. An example here, we have a 4 transcriptional units constructs, 1, 2, 3, and 4. We combine them with BSA1 assembly in a level 1 vector, and we have a 16 transcriptional unit construct. So when I started selecting vectors to adapt my assembly method, I started with pink-green, which is a small vector, and it's suitable to make like large constructs, because when you have like large backbone, you might have issues with instabilities if you are assembling large constructs. However, with the pink-green backbone, I had stability issues. I had transformation issues, especially with the top 10 strain. I couldn't use it to transform a plant cell line, the MM1 line. However, it was good for transforming plant protoplast, Arabidopsis protoplast. Then I moved to PLX vectors, which they have the BBR1 origin of replication. It's a medium origin of replication, which works in both E. coli and Acrobacteria. This backbone was stable. I didn't have any transformation issues. It was good for the transformation of the cell line I was using. However, it wasn't suitable for protoplast transformation because of the BBR1 origin of replication. It was giving like low amount of DNA for transformations. So I moved to the design and development of a new plant vector, which I call PMAP, which it has a small plasmid size, and it uses a new origin of replication, which I made, which is a fusion between the PAC origin of replication from E. coli and the WKS1. This one is an origin of replication from a cryptic plasmid from the bacterium Paracoccus pandotrophus. And this plasmid I found to be stable. It didn't have any transformation issues. It's okay. It's suitable for the MM1 line transformation. And also, it gives high amounts of DNA for protoplast transformation. And I use this plasmid as a core plasmid for the Mobius assembly for plants toolkit. And then I did some testing of the vectors in the MM1 cell line, which is a photosynthetic cell line. You can see it here. It's green, and some cells under the microscope. This cell line was derived from the Arabidopsis thaliana ecotype Landsberg erecta. And it combines both the benefits of plants and microorganisms because it has like faster replication rate than a plant. In about a week, it reaches the that is the steady state of the after the exponential state, and it could be also a good system for iGEM teams, which because like plants are really, they grow really slow, so it might be better to use cell lines. So I tried to transform this cell line by using 4 different Agrobacteria strains: C58C1, GV3101, LBA4404, and NGL1, in combination with the pingreen vectors, the PLX vectors, and the PMAC vectors. And I found that the transformation was Agrobacterium specific. So the LBA4404 strain was giving the higher transformation efficiency, as you can see here. And then GL1 was giving the lowest transformation efficiency. Regarding the binary vectors, pink green always was giving like almost 0 transformation efficiency. And PMAP and PLX, they have like similar efficiency, but only in the case of combining the PMAP with the GV301 strain, there was, I found to be more efficient at using PLX. And then I constructed a library of promoters and terminators for plant expression. So I made like short promoters and short terminators. Until this point, it was really common to use like really large promoters and terminators for plant expression. Sometimes it was up to like 4 kilome bases. The names with the red color is the one I developed in this study. The one with the cyan color, there are some promoters or terminators, they are developed by other scientists, but they were not available in plant repositories. And then black ones, they are like commonly used promoters and terminators. And I characterize these parts in Arabidopsis protoplast. Until then, the characterization of the parts were done in Nicotiana and Bethamiana leaves. And to do the characterization, I developed a high throughput protoplast transformation protocol. In low volumes and 96 well plates. I did scale down to the experiments for high throughput expression, and I use a sensitive and normalized expression system of nano and firefly luciferase. So I had a stable expression of the firefly luciferase, which I was using as a normalizer, and then I had the nano luciferase under the control of a promoter or a terminator, depending what I was characterizing. So I made a characterized set of promoters to fine-tune gene expression. I identify a wide range of activity in the promoters. The ubiquitin genes show high expression, for example, here UBQ11 and 4 in combination with the HSP terminator or NDUFA8 terminator, UBQ11 and UBQ4, they have high expression. And the other genes, the other promoters, sorry, even though they had like lower expression, they are stronger than the NOS terminator, NOS promoter, sorry, which is the commonly used promoter to drive the expression of antibiotics or herbicide cassettes. And also in these experiments, I found that the expression of the promoters in Arabidopsis is different than the activity of the same promoters in Nicotiana and Bethamiana, comparing with existing libraries. Another interesting finding, it was that the terminators highly influenced in gene expression. It's like commonly, it's more likely to use a strong promoter to calibrate the expression, but apparently the terminator has a really influential role in the expression. For example, when I combine the UBQ10 promoter with different terminators, there was almost 5 times range in the expression from 104 to 448 relative units. And the same with the weak NDUFA8 promoter in combination with different terminators. There was a difference of 6.3 in the expression range. And then I did a characterization of the 3 commonly inducible systems that they are used in plants. The index inducible system, the estradiol inducible system, and the ethanol inducible system. In this case, because we have we cannot replace the inducible promoter, we thought that by changing the terminators, we can alter the expression. And in comparison with the constitutive promoters, the inducibles, they show lower range of expression. In let's say there was almost 3 times the range of expression, with the POP6 inducible promoter almost 2. And it was the first time that we did like a direct comparison of these 3 inducible systems. And I found that the Lexay has the highest inducibility. On the other hand, the ethanol inducible system exhibited almost 7 times wide of expression, and it was I found to be leaky in the protoplast system. Another interesting thing, it was there were indications for synergy between promoters and terminators. So a terminator apparently is not always weak or always strong. It depends in which promoter you combine it with, what kind of strength it will exhibit. For example, here, the NOS terminator, it is weak in combination with UBQ10 and POP6 promoters, strong with NDUFA8, and medium with Lexay and the ethanol inducible promoter. APT1 is weak with UBQ10 and NDUFA8, POP6, strong with Lexay, and medium with the ethanol inducible promoter, and some other examples as well. So apparently it matters which combinations you are doing between promoters and terminators when you do your constructs for expression. And then we also deposited a kit in NatGene for for plant expression, there are the PMAP cloning destination vectors, 4 level 1 acceptor vectors from A to delta, 4 level 2 acceptor vectors for, again, A to delta. We have the PLX, 2 PLX vectors, 4 PLX vectors, sorry, with BBR1, which is medium origin of replication, and RK2, which is low origin of replication. 2 for level 1 and 2 for level 2. We have the Mobius universal acceptor vector and the auxiliary plasmids, 12 plant promoters, 13 plant terminators, 3 antibiotic resistant genes, 13 reported genes, which there are like luciferase genes and fluorescent proteins codon optimized for Arabidopsis, and the 3 plant inducible systems. And to sum up, for Mobius assembly, I make this system to have reduced domestication requirements with a minimal vector toolkit to have a high cloning capacity. It follows the phytobrick standard. There is no need for IPDG excal. Currently, there are vectors for E. coli, plants, Cryptococcus neoforms. It's directly compatible with existing libraries. And for the Mobius assembly for plant systems, I developed a high throughput plant product plus transformation protocol, a new plant binary vector, new library of short promoters and terminators characterizing Arabidopsis protoplast. And I found that plant cell culture transformation depends on Agrobacterium strain and binary vector. The terminators influence the gene expression. There is this interesting interaction between promoters and terminators, and also the inducible systems, they don't cross-talk in plants, so they can be used simultaneously in experiments. And I want to finally thank you, all of the people that they help in this work during my PhD, my supervisor, Naomi, second supervisor, Chris, and Gary Locke, who was in my committee, Chris French lab, Marisol who was helping with the experiments, Eric Thoran and Jessica, which they also help in the project. Thanks for listening. Any of you have any questions? I hope I didn't do it like really fast.
Yeah, thank you very much for your great talk. And as always, questions can be written in the chat or you can raise your hand. Yeah, so maybe I have first a question very in interest of our own iGEM project. So we want to do some estrogen-inducing with protoplasts. So we would be very interested in accessing the protocol you have for the high throughput. But
yeah, I do have the protocol. So if you want to use it, just email Naomi to get the okay, and I will send you the protocol.
Yeah, that would be great.
And it's like really, it's really easy to replicate and really reliable. Because I also use some other protocols and I had some problems when I using them.
But it's good to know that there is a protocol that works fine with estrogen, and yeah. Okay, so we have one question from René.
Hello, thank you for the nice talk. I was just fascinated also from the paper you published or the preprint. From the effect of the promoter and termination interaction. Do you know if this effect gets also transmitted if you do it in like stable plant lines? And is it there like, or would you assume that there it's even like more clear or more visible, or would you think that this is more a thing in your transient protoplast assay?
Yeah, we haven't tested yet in the stable transformations. I think Naomi is going to do it at some point. But I believe it's happening as well during the stable transformation. It's kind of a common secret in the plant community that about this interaction, but nobody really talks about it. And also, I think it was found in other systems as well. So I'm not surprised if it's like happening in stable transformation as well. And yeah, I think it has something to do with like DNA looping, like interaction, physical interaction between promoter and terminator, most likely. But it's just like speculations. We need to do like experiments to prove it.
Yeah, I also think like this kind of work is super important, that like people like try or like make the huge effort to like try and measure these combination or combinatorial stuff. Yeah, I think it's great work.
Right, you're right.
Thank you.
Do you know whether this combinatorial stuff between promoter and terminator was also tested to this extent in tobacco?
I'm not aware of like testing it in tobacco. I haven't like been following the development in plant biology the last year because I was working with different organisms. But yeah, I'm not aware of like similar things to tobacco, but probably it's the same effect in tobacco as well.
Yeah, but I find it very, very interesting because in general, people say roughly so that if something is expressed in tobacco, it should also be expressed in Arabidopsis. And
apparently it's not happening like this. I found that the promoters and terminator, most of them, they have like different expression levels in Arabidopsis and in tobacco. Especially the ones that they come from Arabidopsis.
So the Arabidopsis promoters and terminators work different in tobacco, but those who come from tobacco work the same in Arabidopsis, roughly?
I don't know if that happens.
Okay.
I'm not aware of. I know for sure that the ones that they come from Acrobacterium and the 35S promoted, the ones that they come from Acrobacterium, they work like similarly in both chassis, Arabidopsis and tobacco.
Yeah, that's. I find it very interesting. The research you did on promoter and terminator interactions. It's but you don't. So you only have a theory how this difference is made, or were you able to investigate more in this direction? Like on the mechanism, how the difference between tobacco and Arabidopsis, or between the different promoter terminator combinations is made?
Yeah, I assume it has to be with different elements that they are in these parts and how they interact between them and between the machinery of in the two different chassis. But yeah, it needs to be done a lot of work in this area to have like more clear answers.
Yeah, okay. Thanks a lot. So I see no more questions. So I think we can move on to the next point of our schedule.
Yeah.
Thank you very much, guys.
Yeah.
Yeah, thank you for making time to presenting your work.